Good morning, dear students. Uh, Today I'm talking about my second week lecture, and this is my ninth lecture. I'm talking about beta plated sheets and collagen helix. In the last lecture, I talked about alpha helix, then the beta plated sheet and collagen helix. Myself, Mehdi Hayas Shahi, and I'm assistant professor at Interdisciplinary Brain Research Center, Faculty of Medicine, and Aligarh Muslim University, Aligarh, Uttar Pradesh, India. The secondary structure of protein, the beta plated sheet, is a parallel and anti-parallel hydrogen bond geometry and structure examples. Collagen helix, a triple helix structure, glycine, XY repeats, and fibrous protein. Table of the content, which of my today lecture is beta plated sheet overview. In this, I talk about definition, discovery, and biochemical significance. Beta plated sheets, key parameters and examples, dimensions, real protein complex, and mixture sheets. We'll talk about parallel and anti-parallel beta sheet like Haddon bond geometry, polarity, stability, and comparison. Then talk about beta sheet, key parameters and examples like dimensions, real protein examples, mixed sheets. Then talk about collagen helix is overview of collagen helix. Talk example like fibrous proteins, unique triple helix configuration. Then also talk about why glycine XY repeat in amino acid compositions, why glycine is mandatory, and role of proline, apoproline. Collagen, structure and stability, triple helix parameters, cross-linking, and what are the diseases because of the collagen dysregulation of collagens. Uh, the references for this lecture from biochemistry talk about Liberty Strand and also illustrated biochemistry, Harper biochemistry, and also from Lellinger biochemistry. Beta plated sheet is overview is a secondary protein structure. What is a beta plated sheet? Is an extended sheet-like structure. If see this is like sheet-like structure, and it is stabilized by hydrogen bonding. That's very important because many interactions stabilized by hydrogen bonding. And this polypeptide chains adopted a pleated or zigzag conformation, means it has sort of like zigzag. You see here, it's a zigzag conformations. The backbone NH and CO group of different strand form hydrogen bond. Side chain R group alternate above and below the sheet plate, and it's first described by Linus Pauling and Robert Corey in 1951. It's like a sheet structure. You also see this is sheet structure. It is straight, no coil, no turn, and it is 90-degree rotated by 90-degree view of beta sheets, like merge. What are the key characteristics of beta sheets? Also called beta sheet or beta conformation, because in a beta conformation positions, and the phi is minus 120 degree and psi is plus 150 degree. Dihedral angle is very preferred in this beta sheets rotation. And the rise per residue is 3.5 angstrom along the strand axis, 3.5 angstrom. This is the example fatty acid binding protein. Here it has rich in beta sheet. This is the beta sheet, beta sheet, beta sheet, beta sheet. This is all beta sheets. And also it is core structural element in many proteins, like you can say global proteins like immunoglobulins and enzymes. And it is also major component of silk protein, and it is run in anti-parallel beta sheets. Then also it is example of amyloid fibrils, which I discussed last week. Is a misfolded beta sheet in disease in Alzheimer and Parkinson's. And also this beta sheet as beta barrel protein in the outer membrane of porins, as a porins in outer membrane of cells. Now the parallel and anti-parallel beta sheet means it exists in both parallel and anti-parallel. And what is the difference in the parallel and anti-parallel sheets? It has H bond geometry and stability. All strands run in same direction. In the parallel case, there's a one direction, that is NC direction, nitrogen to carbon by to carboxyl direction. A strand connected by long loop region means they are connected by long loop regions, and H bond are slightly bent and oblique. In this case, hydrogen bonds are slightly long, slightly bent and oblique. You see, this is the oblique and bent hydrogen bond. And also, it is less stable than anti-parallel sheets. Stability is less than anti-parallel sheets. And the phi angle is 119 degree and psi is 130 plus 113 degree. Examples: flavodoxine, triphosphate isomerase, barrel protein. This TIM barrel protein contribute almost 10% of the enzymes. Now, what is the characteristic of anti-parallel sheets? Alternating strands run in opposite direction. See, one in this direction and one in this direction is anti-parallel direction. Connected by short hairpin beta turn. So they are connected by short pin beta turn. H bonds are linear. In this case, is very linear. You see, this headed bond is very linear as compared to slightly bent and oblique in the case of parallel beta sheet. It looks like perpendicular to the strand, and it is stable than parallel sheet. It's very stable than parallel sheets. It's very stable than parallel sheets. And the pi is 139 degree and psi is 135 and pi is 139 degrees centigrade. Example: silk fibroin and immunoglobulin. Now the structural dimensions and protein examples. What are the parameters? Phi angle, psi, rise per residue, strand, repeat, hydrogen bond, stability, and loop connections. These are the parameters. And now we compare parallel beta sheet and anti-parallel beta sheets. Pi angle is, in the case of parallel, is 119 minus, and this case is minus 139. Psi angle in the case of parallel is 113 degree, and in this anti-parallel beta sheet has 135. Rise per residue in case of parallel beta sheet is 3.2 angstrom and anti-parallel 3.4 angstrom. And in the case strand repeat is 6.4 angstrom in the case of parallel beta sheets, and anti-parallel studies it's 7 angstrom. And H bond angle is oblique and angled in the case of parallel beta sheet, and in the case of anti is a linear or perpendicular. Parallel beta sheet is less stable and anti-parallel is more stable. Long loop connection in the case of parallel beta sheet and very short beta hairpin in the case of anti-parallel beta sheets. Silk fibrin is example of anti-parallel beta sheet, and immunoglobulin is also example of anti-parallel beta sheet, and Tim barrel is example of parallel beta sheet, and amyloid fibrinosis cross beta structures. This is the Ramachandran plot which gives the beta strand in a range of psi 120, 120 to 170 in this range, and psi 120 to 120 to 170, and the phi is minus 60 range, minus 60 to 110 degrees. Now, the collagen helix is the most abundant protein in mammals. 30% of all mammalian protein by mass, and it is 28 plus genetically distinct collagen exists in the nature, and almost triple helix, that is three polypeptide chain intervened. Glycine XY obligatory repeating sequence triplicate. What is collagen? It is a fibrous structure protein. It is not an enzyme. It's found in the skin, bones, cartilage, tendons, blood vessels, and cornea. Provide tensile strength and structural integrity. Synthesize as procollagen in fibroblast and also osteoblast for the bone. And it is also has extra cellular processing like propeptide cleavage to the tropocollagen. Tropocollagen molecules self-assemble into collagen fibrils. This is very unique characteristics. Tropocollagen molecules self-assemble into collagen fibrils. Why collagen is very unique? Not the alpha helix or beta sheet form its own unique helix. It is left-handed polyproline second helix per chain. Collagen has, and there is three polyproline second helix wind right-handed to form triple helix. Is called triple helix. And glycine every third position is essential. There is no exception in this collagen. Glycine at every third position, and it is essential, no exception. There's a four types of collagen. Type 1, it is mainly found in skin, bone, and tendons. That's most abundant one. Then type 2 collagen, it is the cartilage like hyaline and fibrous in nature. Then type 3 collagen, it is in skin, muscles, blood vessels, especially in the fetal blood vessels also. And the type 4 collagen is a basement membrane or networking forming. The basement give the strength and networking forming. This is the conformation of single strand of collagen. There is a proline here, triple proline here, glycine, glycine, then proline, then again glycine, then proline, and then glycine. This is the conformation of single strand of collagen helix, and this is a cross section of the of the model of collagen. This is a cross section model of the collagen. And this glycine XY repeat and amino acid composition, why every third residue must be glycine, is very important to discuss in collagen. The glycine must be every third position. There's no exception in this case. Every third position is a glycine. Glycine has H at its side chain. This is the smallest R group possible. And this H points into the center of the triple helix. Means it is in the center of the triple helix. Any larger amino acids would cause steric classes, but it is because of hydrogen, there is no classes. A mutation like glycine to alanine cause osteogenesis imperfecta because of the mutation from glycine to alanine. Now the X position. So normally the X position is the proline. Almost 28% of the X position occupied by the protein in collagen and proline's rigid pyrrolidine ring stabilize the pp second helix and create a left-handed helix conformation per chain. Means the helix is left-handed per chain, and it restrict the backbone flexibility. It also important, it restrict the backbone flexibility. and it's become ordered structure. And other residue also found in the X region, that is alanine, glutamine, glutamic acid, also glutamic acid, and also glutamic acid also found in the X chain, and also glutamic acid, and also glutamic acid found in the X position, and also glutamine. Now the Y position, it mainly occupied by hydroxyproline. Hydroxyproline, it is 38% of the Y positions, and 4-hydroxyproline is unique to the collagen and elastin. That's very unique. And hydroxyproline require vitamin C, exactly ascorbic acids, and hydroxy group form hydrogen bond with to stabilize the helix. And hydroxylacin is also at Y position, and that is for glycosylation. It's a site for the glycosylation modification of this amino acid. Now the structural structure and stability, the cross-linking in the disease because of the collagen disturbance, triple helix. This is a space-filling model, shows the collagen. Triple helix parameters. Each alpha chain, left-handed polypurline second helix, 3-chain coiled right-handed around central axis, and pitch per residue is 2.9 angstrom, that is rise along the helix axis, and almost 3.3 residue per turn. Helix pitch is 8.6 angstrom, and diameter is 15 angstrom of triple helix. And the cross-linking and maturation, lysine and hydrolysine residue oxidized by lysyl oxidase form covalent cross-link between collagen molecules, and this cross-linking increase the tensile strength of collagen. Then collagen fibrils then aggregate into collagen fibers. This collagen fibrils aggregate into collagen fibers. Now the collagen diseases because of irregularity in the amino acid of collagen, and clinical relevance, why collagen goes wrong, and genetic and nutritional disorder. Scurvy, well known, a kind of disease that is because of vitamin C ascorbate deficiency. What the mechanism of this deficiency? Prolyl hydroxylase requires vitamin C as a cofactor, and without it, proline and lysine cannot be hydroxylated. There is no hydroxylation of proline and lysine, and it's give the unstable triple helix, and then cause collagen degradation, collagen degradations. What are the signs of scurvy? Is a bleeding gums, poor wound healings, and perifollicular hemorrhage. These are the signs for scurvy. Then Ehlers-Danlos syndrome, EDS. Why it is caused? Because of the mutation in the collagen gene or collagen modifying enzyme. And what are the mechanisms? Defective type 3 or 4 collagen. Defective 3 and 4 collagen. And also impaired cross-linking. There's a problem in the cross-linking, not proper cross-linking. Lysyl, and also because of lysyl hydroxylase deficiency. This enzyme is not present. And the collagen fibrils form incorrectly. And what is the sign of this Danlos syndrome? Hyperextensible skin, hypermobile joints, and fragile blood vessels. These are the signs of Ehlers-Danlos syndrome. Other disease because of collagen's regularities and collagen disease and clinical prevalence. Osteogenesis imperfecta. It is caused because of mutation in COL1A1 or COL1A2 genes. The mechanism: single glycine X substitution in glycine XY repeat. Even one wrong amino acid disrupt entire triple helix, and all or nothing unfolding, and type 1 collagen defective. Sign: brittle bones, blue, sclery, and hearing loss and loose joints because of osteogenesis imperfecta. Means proper genesis of the bones is not possible. That's why it's called osteogenesis imperfecta. Then another syndrome is Alport syndrome, as because mutation in type 4 collagen genes, and that is called 4A3, A4, and 5. The mechanism: type 4 collagen form meshwork of basement membranes, and defective type 4 is a dysfunctional glomerular basement membrane in the kidney. The sign of this Alport syndrome is a progressive kidney disease and also sensory neural deafness and ocular defects. So therefore, the significant that all collagen disease highlights the critical importance of glycine XY sequence integrity and proper post-translation hydroxylation. That's very important. Now there is a comparison of beta plated seeds and versus collagen helix is a comparison. Features this include type of a structure, number of chains, hydrogen bond direction, characteristic sequence, key amino acids, helix handedness, rise per residue, main role, and examples protein and associated disease. Beta plateau extended sheet formation is a very extended, and collagen is a helix is a triple helical fibrous structure. Number of chain, multiple strain in same or different chain, and collagen is always 3 polypeptide chain. H bond between adjacent strain, in this case between chain of the triple helix in the case of collagen. In the case of beta pellet C, there is no specific repeat needed, but in the case of collagen, it's strictly glycine and XY triplet in repeat required. Key amino acid in the case of beta platelet, there is no restriction, all residues possible in beta platelet seed. But in the collagen, glycine at every third position is important, is very important and must. Beta plateted is not a helix, is an extended conformation, but collagen is a left-handed per chain and is a right-handed triple helix. Beta planet had 3.4 to 3.5 angstrom rise per residue. In collagen has 2.9 angstrom helix per residue rise. Beta planet seed shows a structural rigidity in globular protein, and collagen helix shows tensile strength in connective tissue. The example of beta plated seed is a silk fibrodin, immunoglobulin G, and TIM barrel enzyme. And collagen fibril example is skin, bones, type 1, cartilage, type 2. The disease associated with beta plated seeds are amyloid disease, misfolded beta seeds in the neurodegenerative disease like Alzheimer and Parkinson's. In the case of collagen helix, the disease associated with this is scurvy, osteogenesis imperfecta, and also EDS syndromes. Now, the summary and the key takeaway: the beta plated seed extended conformation stabilized by interstrand hydrogen bond, and it has two types exist: parallel in the same direction and anti-parallel in the opposite direction. Anti-parallel is more stable, is a linear hydrogen bonds, and it has pi's minus 120 degree and size 115 degree anti-parallel values. Rise per residue is 3.4 to 3.5 angstrom and repeat is 7 angstrom. Side chains alternate above and below the sheet. Silk fibroin is the best example, is a pure anti-parallel beta sheet examples. And amyloid fibrils is a pathological cross beta sheets. Also, it is found in immunoglobulins and also TIM barrels and porins. These are the example of beta plated sheets. Collagen helix is the most abundant protein in mammals. It is almost 30% by mass. The unique triple helix because not alpha helix, not beta helix. Each chain is left-handed helix. Overall is a right-handed and obligatory glycine XY in sequence. Glycine is non-negotiable, is must and important amino acid for this collagen helix. X often proline, Y often hydroxyproline. Hydroxylation require vitamin C, and if the deficiency is there, this scurvy disease. And glycine mutation cause osteogenesis imperfecta. The cross-linking by lysyl oxidase and is give the tensile strength because of collagen helix. And it is more than 28 types, like skin at first type, then cartilage at second, and membrane at fourth types. So this is a overall summary and take away home the message for the beta for this lecture, beta plated seeds and collagens. Thank you so much.